Let's see. All right. We're back for more genetics. Hold on. I had to go get coffee. And just just so we're clear, the squirrels are back again today and they're annoying the dogs. You will hear them voice their displeasure. Just letting you know straight up. Okay. So, let's walk through a couple things right here. I'm going to go through a bunch of different Punnett squares. And again, today we're still sticking just with Mendelian genetics. So I'm not really going to go into the other types of genetic stuff. On Friday, we'll do some practice work for Mendelian genetics. When I come back on Monday, I'll come back through and review some of this stuff, like in person. So that if you have any questions, you can actually ask them rather than just sort of nodding, going, right, Turner, totally, we totally have this, no worries. Anyway, and then I'll start getting in a little bit more into the more advanced genetics. So to start things off, here we go. Pink. This is going to get all kind of like weird and messed up. So, okay, so we have. I was like really way too thick. We have our Punnett square. Punnett squares, by the way, were not actually invented by Mendel. The dude's name was Punnett, but I can't remember his first name. I'll come back to that. Anyway, okay. So one of the easiest ways that we can sort of really initially see how a Punnett square is set up is sex determination. So females are XX, males are XY, and XY. There we go. I'm just like I want. I'm doing this mostly for me, I guess. Anyway, so in this case, remember we're only talking about chromosome pair number 23. Chromosome pairs number 1 through 22 all look like XX. They would, at least after they go through the S phase of my of yeah of mitosis, then you wind up with four sister chromatids instead of just like two. It looks like an X. Okay, so females, that last chromosome, they both look like X's. Males, they look like a Y. Males are physically missing a portion of their DNA. Females have more DNA than males do. A lot of women think that that makes them superior to men, which is probably true. Anyway, I'm going to get drawn off the subject on that one. I'm not going to let myself do it. Okay. So if we have a female, okay, due to independent assortment, this female is going to make two eggs, and one of those X chromosomes is going to go into one of those eggs. The other X chromosome is going to go into the other one. With the male, an X chromosome goes into one sperm, a Y chromosome goes into another sperm. And I won't get into more than that. Anyway, so what we're trying to do here at the Punnett Square is to just show the distribution of that. So all a female can actually donate is an X chromosome. Males are the ones that actually determine whether it's going to be a girl, XX, or a boy, XY. So if the male donates the X chromosome, it's going to be a girl. If the male donates a Y chromosome, it's going to be a boy. About 50-50 chance of between a boy and a girl. And if we look at just ratios of male versus females, they come out to about 50%. I think if we look at like global population, it's like 51% female, 49% male, but that's mostly because females tend to live longer than males. We're not going to get into the whole superiority thing anymore. Don't. Shut up. So it's the male that determines the baby's sex. Now, you guys remember King Henry VIII? Or is it Henry VIII? Yeah, it was Henry VIII, right? I don't know why I'm asking you. Anyway, who like kept killing his wives because like none of them would give him a son. Like it was at Anne Boleyn, and that's the only one that I can remember. Anyway, yeah, he keeps killing off of his wives because they're not giving him a son. Yeah, it was his fault, wasn't it? It's all any of those women could do is donate an X chromosome. He was the one that did. I guess if I were to read my own notes, then I would see that it actually says Henry VIII. Okay, so I walked through yesterday and talked about the distribution of genes in a Punnett square, and let's talk about this just real quick here. If we have a heterozygous trait, actually, you know what? Let's just move on, because I want to talk about all this stuff anyway. Okay, so there we go. Now you guys can actually read it. We have a couple of different ways that we can look at allele distributions. Okay, so remember that we have two different letters, and what those represent is dad donates one letter, mom donates the other letter. Okay. In the case of Mendelian genetics, this is a complete dominance type of thing. So if a dominant trait is expressed, if a dominant trait is present, dominant trait is expressed. So here what we have is two big A's are homozygous dominant. Homozygous means they're both the same. If we were to have, there we go, big A, little a, meaning dad donated a dominant allele, mom donated a recessive allele, or vice versa, it really doesn't matter. Whoa. Sorry, hang on a second, guys. I'm running into technical difficulties here. All right, let's try this. I know you guys are like, what are you doing? This is the only way that I can actually get a camera to actually work on here. There we go. Okay. I don't know, weird, right? So this big A, little a is actually the same as little a, big A. It really doesn't matter. They actually mean exactly the same thing. We always tend to write it, or at least I always tend to write the dominant letter first, the recessive letter second. It doesn't have to be. These are variables. It really doesn't matter, but I find that the more consistent you can be, the better that works out. So we're not going to do that like that. So if you've got one dominant, one recessive, this is heterozygous. And then if we have both recessive, technically that's homozygous. A lot of times we actually don't even refer to it as anything, because if it's homozygous recessive, if a recessive trait is expressed, that's the only way that it could be. Like you couldn't have any dominant trait that's actually present. So it's just kind of, we just typically say recessive. So it's like whatever. Okay. Then we also have genotype. Stop it. Shouldn't have said that. That was my wife. Anyway. Genotype are the genes involved. So that's typically like the letters. Phenotype is the physical traits. I think we covered this yesterday. Awesome. The awesome trait, right? So phenotype is red hair. Hi. Brown eyes. Hi, puppy. Okay. Oh, thank you. Okay, I have to get back to work. Okay, get down. Come on. Yeah, get down. So phenotype, here comes the jealous dog. Like, get away. Is the physical traits. Okay, so let's look at. Let's change the color of this. Let's look at the Punnett squares. So you can distribute these. That's terrible. It's not so good either. Even when I have a tool helping me draw straight lines, it doesn't come out straight. That's awesome. Okay, so again, with the Punnett square, we can actually sort of distribute this stuff as we go. But here's the thing. With Mendelian genetics, when he first looked at pea plants, I think we had like pod shape, like the pea pod shape. We had pod color. Pea shape. So like pod shape, it could be smooth or it could be constricted, where like it's like you can sort of see the peas through the pod. Pod color, it can be yellow or green. Pea shape, it can be smooth peas or wrinkled peas. Pea color could be green peas or yellow peas. Here's the weird one. Yellow is the dominant. Green peas are the recessive. Basically, can you imagine getting like yellow peas in like the grocery store or something? Like people would freak out. But that's the dominant trait. I don't know. I guess we just only grow the recessives. Hi, puppy. We have plant height, flower position, two, three, four, five, six. Oh, and flower color. So plant height, you can have tall or short. That's genetic. Flower position, terminal or axial, meaning did the flowers come out from like the main stem? That would be axial. Or do they only come out from the top? That would be terminal. And then flower color, it's either purple flowers or white flowers. Okay, so those are like the different traits. And remember yesterday I was telling you that Mendel got like really lucky when he was looking at peas, because each one of these traits is only controlled by a single gene. Only one gene. That's cool. That makes things a lot easier. Now, you probably remember like in honors biology going through and doing a whole bunch of like practice problems. And we're going to do some of those. But here's the thing. There's only 4 ways of doing a monohybrid cross, a cross looking at one trait. There's only 4. Actually, there's 6. I guess I lied to you again. It's a habit. I just can't help it. But you're only going to see 4. So here are the genotypes of the 6 different ways that we could do this. You could have, we'll do T's. Big T crossed with big T, big T. You could have big T, little t crossed with big T, big T. You can have big T, little t crossed with big T, little t. You can have big T, little t crossed with little t, little t. And let's see, What else am I missing? Big T crossed with little t, and you can have little t crossed with little t. Okay, so I told you that like there's really actually only 4 ways. There's 6 of them up here, but here's the thing. I'm never going to give you that. Because if you cross a homozygous dominant with a homozygous dominant, you know what you're going to get? You're going to get homozygous dominant. And I'm never going to give you that. Same reason. So there's only 4 ways of doing a Punnett square. If you know how to do those 4 ways of doing a Punnett square, you're golden. I know. Okay, so let's look at one of these. Big T, little t crossed with big T, big T. Okay, so when we do this Punnett square, we're going to look at a couple of different things when we're done. We're going to look at the ratios of the genotype and phenotype. So we do big T, big T, big T, little t, big T, big T, big T, little t. Okay, so now we're going to look at the genotypic ratios, meaning how many are big T, how many are big T, how many are little t. All right, well, there's 2 of them that are big T, 2 that are big T, and 0. So the genotypic ratio is 2 to 2. Okay, now we want to look at the phenotypic ratio. Let's say that this is representing tall versus short plants, if tall is the dominant trait. By the way, I didn't mention this before. We typically use the letter representing the dominant trait. Not always. You can use T's for every single genetics problem that you want to do. It doesn't really matter. It's like X's and Y's in algebra, whatever. So in this case, we want to know how many of these are tall, how many are short. Now, in order to be short, it would have to be completely recessive. It would have to be little t. So how many are tall? 4 of them. How many are short? 0. So the ratio of tall to short is 4 to 0 or 1. We could also look at like percentages, what percentage is whatever. So sorry, I know this is taking a while. Okay. We'll do this one here. This is sort of the classic heterozygous. So we've got big T, big T, big T, little t, big T, little t, little t, little t. The genotypic ratio, how many of them are homozygous? 1. How many are heterozygous? 2. How many are homozygous recessive? 1. The genotypic ratio is 1 to 2 to 1. The phenotypic ratio, how many of them are tall? 3. How many of them are short? 1. That's it. Those are Punnett squares. Okay. Oops, let's do this. One more. This is the last one, and this is sort of the goofiest one. Dihybrid crosses. Everybody loves dihybrid crosses, right? So let's say that we had a heterozygous tall heterozygous round peas rather than wrinkled peas. Okay, so we've got 2 traits that we're looking at here. We've got heterozygous tall, heterozygous round. We're going to cross that with a heterozygous tall and wrinkled peas, meaning completely recessive. All right, back in biology, we would have to figure out, all right, so this one is going to go up here. Change the color. This one will go down the side. And we have to sort of move all these different things around. Okay, so we'll do the first T with the first R, big T, big R. First T with the outer R, big T, little t, big R, little t. You guys remember doing these? I hope that you remember doing them. I hope that you remember how miserable everybody was when we were doing these things, because this is stupid. Doing these types of big dihybrid crosses is annoying and it's ridiculous, and you don't need to know how to do it. You will never have to do something like this. That's not to say that you won't have to do dihybrid crosses, but I have a better way. And that better way is doing, there we go, 2 monohybrid crosses. All you have to know how to do are monohybrid crosses. I'm telling you, this is not going to be hard. So let's do the T's first. Okay, so this one is going to be the T over on the left-hand side. So we've got big T, little t, and then if we look at the other plant, that's also big T, little t. Okay, we've seen this one before. Big T, big T, big T, little t, big T, little t, little t, little t. Okay, now let's look at the R's. We've got big R, and then a little r, little r. So there we go. So whenever we're doing a dihybrid cross, the question is always, all right, what is the possible outcomes that we could have, and what are the potential percentages that you would see? You know, it's if we're just looking at the tall trait here, if we're just doing a monohybrid cross, we'd say, all right, what are the chances that if you cross these two plants together, you'd have offspring that are tall? Well, it's about 75%. It's 3 out of the 4. That's easy. When we're starting to look at 2 traits, we need to first ask our question. All right, so what are the chances that the offspring, let's say, will be heterozygous tall, heterozygous round? Okay, here's the thing. You can do that huge Punnett square, which the chances are you're going to screw one up, or at the very least it's going to take a long time. Or you can just do 2 monohybrid crosses and then multiply fractions. So how many of the T's are heterozygous? There's 2 of them, or 1/2. How many of the R's are heterozygous? There's 2 of them, or 1/2. So the chances when you do this dihybrid cross that they're going to come out heterozygous for tall and heterozygous for round is 1/4. That's it. That's all you got to do. Let's do another one. I don't mean another one. Okay, so let's say, what are the chances that it will be short and wrinkled? Okay, so how many over here are short? That's 1/4. How many over here are short, or I'm sorry, are wrinkled? That's 1/2. So 1/4 times 1/2 is 1/8. That's it. I don't know why we actually taught like that huge dihybrid cross, because it's confusing and it screws everything up. We could do a 3rd trait. Do you think that if I added in big A, and little a, could we do a trihybrid cross? Yeah, that's freaking easy. All you have to do is do another monohybrid cross and keep multiplying the fractions. We'll come back to that. We'll do a little bit more on those. Tomorrow, we're going to do some Mendelian genetics practice, and I'll come back and I'll show you these things again.